So in the last slide, we were discussing on the digestion of proteins, right? We have started with the digestion of proteins, and we have already mentioned about the various sites where the digestion is carried out. The salivary gland or the oral cavity do not involve the digestion of proteins because there are no enzymes which are able to carry out the breakdown of the peptide bond, right? So the protein components of the dietary sources will be remaining intact, and they will be entering into the gastric cavity where the digestion is initiated, because peptide bonds are very strong bonds, they are covalent bonds. So these proteins were to be first subjected for a denaturation process, and this denaturation is carried out only in the gastric cavity with the help of concentrated HCl, which is present in the gastric juice. So gastric juice contains a very important component known as concentrated HCl, which carries out the digestion of the proteins. So the function of the HCl in the digestion of protein is happening in three different ways. First, it maintains the pH of the gastric cavity in between 1.6 to 3.2, because we know that the proteolytic enzymes are all synthesized and secreted in the zymogen forms, because they are very reactive enzyme molecules or protein molecules which can even carry out the digestion of the intact proteins, the body proteins itself, unless they are processed in the proper manner. Okay, so these zymogen molecules or the zymogen forms of the enzymes has to be first converted into the active form, and pepsinogen is the first the peptide enzyme or proteolytic enzyme to come into action for initiating the digestion of the proteins. So pepsin is actually synthesized and secreted in the zymogen form or the inactive form, which is known as pepsinogen. So the conversion of pepsinogen to pepsin is the first step for carrying out the denaturation of the proteins or the catabolism of the proteins. So how is the pepsinogen converted into pepsin? The pepsinogen is converted into pepsin only at a pH of 2. So it is the optimum pH of pepsinogen to get converted into pepsin, and this pH of 2 is actually given away by the concentrated HCl. So that's one of the main function of HCl, that is to provide a pH of 2 so that pepsinogen can be converted into pepsin. Again, we can in last slide we have come across the fact that pepsin is a carboxyl peptidase, that is carboxy protease, that is its active site contains aspartic acid residues. So the active site it's an example for an aspartate protease. There are different types of proteases based on the type of the amino acids or based on the type of molecule which is present on the active site. For example, metalloenzyme carboxypeptidase A, which is present in the intestinal juice, is pancreatic juice, is a metalloenzyme because it requires zinc. Zinc is present in the active site, so that is the zinc is always there. It's not coming into and getting bound to the active site at the site of action or at the time of action. It is already there. So carboxypeptidase, the structure of carboxypeptidase gets completed only if the zinc is present in the active site. So it's an example for an enzymatic metalloenzyme carboxypeptidase, right, which is present in the pancreatic juice. Similarly, there are majority of the protein degrading enzymes or proteolytic enzymes are serine proteases, which means that those proteases or proteolytic enzymes will be having serine in their active site. Another example is aspartate protease or carboxy protease. An example for carboxy protease or aspartate protease is pepsin, because pepsin is having carboxy. We know that carboxy contain or dicarboxyl amino acid, aspartic acid and glutamic acid, they are having more than one number of carboxyl groups, so they are named as carboxy amino acids. So they will be having more than one number of carboxyl group, and examples are aspartate and glutamate, and the aspartate residue is present in the active site of pepsin. Okay, so hence it's belong to the class of carboxy proteases or aspartate proteases, which means that those enzymes which belong to the class of aspartate proteases or carboxy proteases are having aspartic acid or glutamic acid. In the case of pepsin, it is a aspartate protease, which means that aspartic acid is the amino acid which is present primarily in the active site. Of course, other amino acids are also there, but the primary amino acids which is present in rich quantities in the active site of pepsin is aspartic acid. Okay, so there are two aspartate residues in the active site of pepsin. Okay, so that is a function. So pepsinogen has to be first converted into pepsin, and this conversion of the zymogen form to the active form is actually mediated at a pH of 2 and is provided by gastric HCl. Again, another function is that throughout the gastric cavity, the acidic pH is maintained, right? Because the maximum pepsin activity has to be carried out. Pepsin has to be has to carry out its function at the maximum level, and this is maintained again by providing a pH environment between 1.6 and 3.2. That is the second function of HCl, and one of the another important functions that HCl itself is an enzyme. I mean, it's a compound which can carry out the denaturation of proteins. We know that what is a denaturation. What do you mean by denaturation? It is a destruction of the higher levels of organization of proteins. That is, if a protein is having quaternary structure or tertiary and secondary levels, all which are made up of non-covalent interactions like hydrophobic bonds, ionic bonds, van der Waals interactions, etc. So all these weak bonds are broken up at high temperature or in the presence of acids, alkalis, etc. So this phenomenon is known as denaturation. So the denaturation process again enables the pepsin to carry out the digestion in a rapid manner. So that is the third important function which is carried out by HCl. Okay, so one is the provision of optimum pH for the conversion of pepsinogen to pepsin. And once a few pepsin molecules are formed, more and more pepsins are synthesized with the maintenance of pH in between 1.6 to 3.2. That's the second function. The third function is the denaturation of the protein, so that the higher levels of organizations are dismantled or it is destructed and uncoiling, so that is it's converted into a linear molecule. That's the primary structure. So the primary structure of proteins are produced. So that is how the partial digestion of pepsin is being carried out in the stomach. Again, pepsin is an endopeptidase, so it can act deep within the molecule. Okay, and pepsin can also catalyze the denaturation of milk, that is the coagulation of milk can be carried out by pepsin. So that again casein, the protein present in the milk is casein. You might have performed the practicals in the first year. So casein is the milk protein, and this casein will be, it's a sulfur containing protein, right? So this sulfur containing protein is undergoing denaturation by the activity of pepsin. So milk undergoes coagulation, so that casein is converted into para-casein. The denatured protein is known as para-casein. This para-casein, what is the next fate of para-casein? Para-casein gets clubbed with calcium ions, which it reacts immediately with the calcium ions, so that it is getting precipitated out in the form of calcium para-caseinate. It is nothing but the curd, right? It is so the casein will be converted into a whey protein known as whey protein. That is, casein loses its natural consistency, so that it undergoes denaturation to give rise to a compound known as para-casein. This para-casein immediately gets clubbed with calcium. It reacts with calcium to give calcium para-caseinate, and that is known as the curd. You might have seen in the newborns, they will be having only breast milk as their food, right? So immediately after being fed with the breast milk, we will that is in order to remove the gas. So you can see that they will give away some sort of white curdy thing will be getting vomited out, right? That is nothing but the para-caseinate, calcium para-caseinate. Okay, so that is the thing. That's how the digestion of milk is happening. So digestion of milk happens in the stomach, right? And from that, only the majority of the milk which is present in the dietary source will be undergoing digestion in the completely in the stomach itself. The remaining will be undergoing digestion in the intestine, right? So the partially digested protein, which is known as, we call it as chyme. So this chyme now enters into the small intestine, right? And on entering into this small intestine, the chyme, what happens? The pancreatic juice has to be secreted. The pancreatic juice will be secreted. But again, we have told that this the proteolytic enzymes are all what they are all highly hazardous substances, because if they comes into contact with our intact proteins, that is, body itself is made up of proteins. So if somehow these proteins, this pepsin, come into contact with the gastric cavity is made up of cells and proteins, right? So what happens? This gastric cavity can undergo destruction, but it's not happening. Why? Because the gastric cavity will be protected against this corrosive action of this pepsin and HCl with the help of a compound known as mucin. There is a thick layer of mucin which is gelled by HCl. So this mucin actually helps it acts as a coating on the surface of these gastric cells, thereby protecting it away from the action of the corrosive action of the pepsin and the HCl. And the main site of action of pepsin or peptide bond ഉണ്ടെങ്കിൽ, which are the sites where the pepsin go and get destructing the peptide bond, wherever it meets the tyrosine residues, the phenylalanine, the tryptophan, the leucine, glutamic acid, and glutamin, right? So these are the amino acids which are subjected to action by pepsin. That is, wherever it is where pepsin is encountering all these amino acids, those peptide bonds will be broken up by the pepsin. So pepsin is not having other actions on other amino acids. It mainly acts on the amino acids tyrosine, tryptophan, phenylalanine, that is aromatic amino acids, and also leucine, glutamic acid, and glutamin. So these are the amino acids primarily which is subjected for the proteolytic action of pepsin. Okay, so that is how the digestion is happening in the stomach. Now, what is happening in the intestine? So peptic digestion will be producing the partially digested products. And what happens? These partially digested products, when enters into small intestine, it results in the release of a hormone known as cholecystokinin pancreozymin from the duodenal mucosa. From the duodenal mucosa, the entry of the chyme or entry of this partially digested food into the small intestine will result in the release of hormone known as cholecystokinin pancreozymin, right, from the duodenal mucosa into the blood, and this results in the stimulation of secretion of pancreatic enzymes, so that the pepsin helps to initiate. So another function of pepsin is to initiate the digestion or to continue the digestion by the pancreatic juice in the small intestine. So once these partially digested food enters into the small intestine, it is coming along with pepsin. So as you move down the alimentary tract, you can see that the acidic pH will be getting replaced with alkaline pH, right? So once the small intestine is reached, as you descends down the small intestine, the alkalinity increases, and this alkaline medium causes the denaturation of pepsin. So once the food enters into the duodenum, slowly the action of pepsin will be stopped due to the alkaline pH present there, right? So that is how the pepsin is terminating its action when it is entering into the duodenum. Now, what is happening in the duodenum, or what is happening in the first part of the small intestine? So, when the partially digested food, when enters into the duodenum, the first portion of the small intestine, what happens? This pepsin itself causes the stimulation of duodenal mucosa to release a hormone known as, there are two hormones, cholecystokinin and pancreozymin. So, these two hormones catalyzes the synthesis and secretion of pancreatic juice into the small intestine. Now, what are the things present in the pancreatic juice? As we know, as we all know, that is, the pancreatic juice is very important for the digestion of all the biomolecules, because the main digestion is carried out by the pancreatic juice, regardless of whether the biomolecule belongs to carbohydrate, the lipids, or the proteins, right? So the main digestion is carried out by the pancreatic juice. So the contents of the pancreatic juice is very important, right? So in the case of proteolytic enzymes also, the pancreatic juice contains very potent enzymes, which includes trypsin, chymotrypsin, proelastase, and carboxypeptidase. So these are the main four enzymes present in the pancreatic juice for carrying out the digestion of proteins, the partially digested proteins or the proteins, right? So trypsin, chymotrypsin, proelastin, and carboxypeptidase, all of which are synthesized in the zymogen forms.